So welcome, good afternoon, and my name is Karambod Siddiq, Director of the UWA Institute of Agriculture, and I wish to acknowledge that the university is situated on Noongar land, and the Noongar people remain the spiritual and cultural custodians and continue to practice their languages, beliefs, and I'm a yes voter. So make sure that you vote on, is it 24th of October? Something like 14th, yeah. So on the behalf of the University of Western Australia and Institute of Agriculture, I welcome you. I think we got a reasonable good number for this special lecture delivered by Adjunct Professor Madan Pal Singh. So he's also an adjunct with us within the Institute of Agriculture. Professor Singh is currently ICAR, the Indian Council of Agriculture Research, Emeritus Scientist, Plant Physiology, at the Division of Plant Physiology, Indian Agriculture Research Institute. So a lot of people here have been there. It's one of the most prestigious agricultural postgraduate education research institutes in India and also globally. His work has been on high temperature tolerance in many crops, including legumes, but more recently has been focusing on rice. Of course, prior to this position, he was a professor and principal scientist at the Division of Plant Physiology and guided a number of masters, 30 master's students and equal number of PhD students. And Professor Singh contributed as a physiologist in the development of 11 varieties of important crops, including chickpea, 4 lentils, 2 mung beans, 1 basmati rice, which is very famous for drought stress tolerance, published 173 research papers in peer-reviewed journals, and edited, co-edited 5 to 6 books there. I also know Madan has worked very hard to get the Indian Society of Plant Physiology, which is one of the prestigious societies, Neil, you know, and that society has really progressed well. Their journal called Indian Journal of Plant Physiology, which is quite a good journal, had no impact factor because that's the way things operate. So Madan and myself and few others got together. We changed the name now called Plant Physiology Reports, and then we commissioned Springer to help us, and with hard work, now it got its first impact factor this year, just over 1. Yeah, so I think that journal will charge, and so make sure that some of us publish some papers in that journal. And Madan also has been very, very kind when we had the COVID-19, and 2 of our students were positioned there, and Sneha is not here today, and Agai is in the field. Those 2 PhD students, one working on drought tolerance in chickpea, one on heat tolerance in wheat. They were positioned there. Madan and colleagues, including Bharadwaj, looked after those students. All their experimental expenses all were met by IARI, and Sneha has submitted 1 paper now, and Agai is working on the paper. So thank you for looking after our students. Now the students are back here, the COVID is over, and they won't be going back to India for the experiments. They will do it here. So Madan is going to talk about phenotyping for rice, high temperature and CO2 environment. The facilities in IARI is one of the best in terms of phenotyping, and he will show some of those facilities. Thank you for coming us, and welcome, Madan. Thank you. You have to use page up and down, and sure. This one you can use for your thing. That'll okay. Thanks again. Yeah, sure. Cheers.
Thank you, Professor Kadambot, for the nice introduction, and also I'm thankful to you for your kind invitation to UWA Institute of Agriculture here, and giving me the opportunity to interact with the faculty and the students of this institute. So good noon, everybody. I hope you had the good lunch. I welcome you all to this my seminar. I would like to share you some of the work which we have been carrying out here in rice. Basically, the other crops I not include because it's not possible to include everything here. So in brief, I am going to share you some of the work on the rice. So this is the topic, the phenotyping of the rice for the high temperature and high CO2 environment. So before going into my presentation, just I will give the brief background why we are focusing this type of work in the rice. You know that the CO2 temperature and water availability are the three important metabolic drivers of the plant system. And under the climate change scenario, all three are really going it's okay. Yeah. So all they are going to change the current climate change scenario. You can look into the current CO2 level has been estimated to be 422 in the June 2023, and this CO2 is very important considered for the C3 plants, and people have already projected that the C3 plants can have the better advantage. So here I have taken the very old picture from the literature of the rice, where the one C3 and one C4 plant have been exposed to the different level of the CO2, and they have found here very critically that the response of the C3 plant, the rice here, you can look into the CO2 level 300 ppm and the 400, and then this is the highest level is 800 ppm and the control is 390 ppm. So you can see that response visibly, the growth response of the C4 C3 plant is very clear compared to the Citeria viridis, which is C4 plant. Even at 100 200 ppm there is no much difference in the level of the treatment of CO2, and there is a almost same response. So we can say that C3 plants are going to respond better, and how they are going to have the advantage, they can have the better enhanced carbon assimilation, water use efficiency, and the main effect comes through the suppression of the photorespiration. These are the some of the theoretical aspects. Coming to the scenario of the projections about the high temperature, this is the data of the last 140 years, already known that the level of temperature increase. About the projections globally, they are the 1.8 to 4.0 by the end of this century. Then for Indian region, particularly South Asian region, it has been projected differently between 0.88 to 3.16 by 2050. And between 1.56 to 5.44 by 2080. And this year, currently last month of the August has been really witnessed as the most warming August month in the last 15 years in India. And the temperature of August, which is really for the rice, is a very important crop which is grown in the North Indian conditions, which is known the rice bowl of the country. The temperature has been 37 or 38 degrees Celsius, while the optimum the temperature for the rice is 30-31 degrees Celsius. You can understand how this can be affected, and even in 2017-18, the government of India has once and given a warning that temperature may be up by between 1.7 to 2.2 by 2030. This is the background. Then how the CO2 and temperature can affect the plant. So it is just overall response. People think that when the CO2 assimilation can be better in C3 plant, they can really have the high advantage and the grain yield can be increased. But CO2 is not only increasing, it is the temperature also concurrent increases in both the factors. So what people have, this is just a calculation only, that once we have the higher photosynthetic activity is there, because of that the optimum PR level, but because the increase in temperature, the rate of respiration is a very important factor which is increasing. So we can see that effect of high CO2 in the photosynthesis will be only a particularly small window of the time, but the temperature will affect the rate of respiration throughout the 24 hours, maximum may be there in the same time. So what is estimated based on this gain in assimilation, we are expecting higher photosynthesis, maybe the increased biomass and grain yield, but there will be higher respiratory losses will be there. Another important impact can be there, the rapid respiratory development of the crops will be there. So it is an estimated that the net gain cannot be more than 5 to 7%, while the yield response people object up to the 20 or 25%. So with that one, this is the how the climate change can affect our food crops. Already yesterday, Professor Siddiq has mentioned that 786 millions are hungry every year, and they require so we need to increase the food productivity by 50% to meet the demand of our population in the worldwide. Also the climate change mediated is temperature rising and flooding and so many other extremes are there. They can also affect the productivity already projected. So but the temperature level, when it is increased along with the other frequency of weather extremes, it could be very detrimental for the rice crops. I am focusing on the rice only. So looking into these scenarios and also the impact, what could be the best ways to combat these climate related factors are there. So in our we have focused on some of the activities in this area in the rice. So these are some of the experiments we have conducted, starting from the phenotyping of the rice, exercise for the reproductive stage, high temperature stress tolerance. So why we are focusing on the reproductive stage, I will go to the next slide, I will explain to that one only. Then analyzing because the CO2 is also is known to be very positively affected. So we have taken the last set. So this much of number 240 and 250 number is very large, and to have to know the impact of such a large number of the genotypes in the OTCs and phase system normally used for CO2, really it is not possible. So we have used one of the plant geometry technique, which is basically standardized in the IARI people, and for the screening the large number of the genotypes, and this is based on low plant density. Details I will be sharing the next one. Then from this one, because based on simple plant geometrical technique, you cannot rely it until unless you have validated the same genotypes under the real high CO2 environment. So what we have taken the 23 of some of the genotypes, and then we validated their response under the phase enrichment system for the CO2. And lastly, we have taken a brief study regarding the impact when the CO2 and temperature together they are given to the plant system. We have taken the two contrasting genotype, one is the tolerant type, one is the sensitive type, and then we have studied their response, how they are going to behave when temperature as well as the CO2 are then higher. So this is the briefly. Coming to the first one, why we are going to focus on the reproductive stage tolerance. You can see here in the first slide, it is also the report from the ED study. You see that this is if the anthesis stage is shown as a 0, if the high temperature exposure plants are given at the anthesis stage only, the day of the anthesis, you can see large reduction in the fertility occurs, and we have only just 20%. On the other hand, if you take the 5 days before the anthesis, the high temperature exposure is given. So it is only having very little, nearly say 70% fertility is there. So we can see here that the anthesis stage is quite sensitive in the rice, and if you are going to have the tolerance level improvement approach for the rice, we should focus on the reproductive stage. And this is the data again from our 10 years data average is there from one of the ACRIP project of ICAR, and from the North Indian conditions they have averaged the temperature. So what you can see here that during the season of the creep, when we have the crop from the up to 150 days by November, so this the duration between the 80 to 90 or 100 days, it is basically coinciding the flowering stage. So here in the Northern Indian conditions, the temperature maximum of the day, it is nearly approaching the 35 degrees Celsius, the average temperature. So it is also a alarming situation. We have to look into this aspect very seriously because it can have the large impact on the rice productivity in our North Indian conditions. So this is a slide. So for the rice crop, it has been estimated the temperature about the 35 degree, it can negatively affect all stages of the growth and development, and it is considered detrimental to fertilization mode. Then a rice line, how we can say that it is tolerant one? If a rice line is capable of maintaining more than 70% spikelet fertility, because spikelet fertility is one of the critical trait to identify any genotype for the high temperature tolerance. So at the temperature exposure above 35 degrees Celsius during anthesis, if it is having more than 70% fertility, then we can consider that it is one of the heat tolerant type. Okay. And during the last 1 decade, we have screened nearly 922 rice accessions for the flowering stage high temperature tolerance, and we have identified some of the donors, and some of them are being used for the breeding program also for the heat tolerance development. So this is one of the study. This work we started in the year 2011. That at the time the facilities were not there to particularly expose the plant, only the flowering stage only. So what we have planned, we have taken a initially set of the 71 genotypes, and most of them representing Nerica, that is the new rice for Africa, Nerica type germplasm was there. So basically the Nerica lines are there developed for the heat tolerance, but we wanted to phenotype them. What is the response there for the heat tolerance? So normal time the temperature cannot be that much high. At that time it was not observed. So what we planned, we planted the rice in the we have pre-planted the rice in the month of the March, so that the idea was there, these plants can have the coincidence of high temperature in the month of April at the seedling stage, and the flowering will occur after the 60 or 70 days, maybe it may be coinciding by the June month. So the high temperature exposure more than 35 can be there. So what we observed, when the flowering was there, the temperature at the vegetative stage, it was nearly 43 degrees in the month of May. So because of the high heat stress of the 43 degrees Celsius, out of 71 the field condition, you can say some of them they died, they could not survive. So out of that only 23 could survive under the heat stress. So we could screen them that these 23 are at least the vegetative stage tolerance is there in them. Okay, so an interesting thing was there that one of the line, you can see the N22 is an international check for the drought stress tolerance. And this in N22 also showing the scorching, the heat margin, the leaf margins were showing the scorching symptoms of heat. But on the other hand, this line one on Nerica L44, Nerica L is for lowland 44, it was found to be very tolerant. There was no effect visibly seen on the growth. Same way you can see the response in the flowering and panicle stage. So this is the just so what we did initially, we had done the field screening. Then we have taken the 3 of the contrasting genotype, one the N22 and the Nerica L44, and another is the mega variety IR64. So these 3, but from the initial field screening, then we had grown into the normal season of the crop, and we have covered them under this type of polychambers. and inside the polychamber the temperature also raised up to by 37-38 degrees Celsius. Then the same genotypes were confirmed again by growing them inside the growth chamber only, because the precise control can be there in the growth chamber only. So this is the data. You can see how the in the all the three experiments, the interesting part is here, the Nerica L44, it is having nearly in the this study, in the first one it was 85% fertility is there. When it was conducted along with this polychamber, here also it was able to maintain, and same it was at par in the fertility with the N22. Same way when we grown them in the chamber inside, you can see NL44 at the high temperature stress, it was slightly lower, but nearly 70% fertility was maintained. So these all studies, they confirmed them. And then also another study was there, the leaf surface temperature, because the cooling effect is very important right here. So what you can see, leaf surface temperature of this our NL22 is there nearly this is the ambient air temperature was this one, and the leaf chamber A is the chamber air temperature. Inside the outside temperature was nearly 33, and inside the chamber temperature was nearly 38, but this the leaf tissue temperature was maintained by 34. So this shows that how these genotypes are able to maintain that tissue depression is there, so lower temperature is there. Then another important feature here, this is for N22, you can see here. So if they are able to maintain the control their tissue temperature, we can say they can avoid the heat stress effect. Another way is the escaping. You can see here, this is the, we have recorded the number of florets flower or the anthesis occur at what time of the day. So you can see here, this was the at between 9 to 10 a.m. how many number of spikelets show the flowering or anthesis occurs or the anthesis burst occurs. Then between the 10 to 11 a.m. and this is the time you can see 10 to the heat stress start after 10 o'clock, and then you can see the 11 to 12 and 1 p.m. So N22 maximum floret the flow anthesis occurs in the N22 because between the 9 to 10. So the anthesis in this one it shows that the N22 is escaping type, it escapes the heat stress effect. But this NL44, it is showing the maximum peak, and the same way we also found in the IR64. So it shows the maximum flowering, so it is not trying to avoid. So it is having the true tolerance, because the flowering stage is the main critical stage, and at that time it is showing the maximum flowering here. So it shows that this variety is really having the better tolerance, this line having the tolerance, it can be used as a source of the for the plant breeding program. Apart from these physiological traits, we also found the higher photosynthetic rate in them. Also some of the antioxidant enzymes were there, and we found that under high temperature and humidity, their activity was induced higher. Then another, so based on these are the, so based on the this line after the, because it has a higher survival percent at the early stage, so we found that it has a vegetative stage tolerance. Then based on the high spikelet fertility, we can say that it is also having the reproductive stage tolerance. Then the other excellent, it possesses the excellent agronomic traits like the grain yield and head loss recovery. So these are the important three categories. So considering all these levels, it was found that this Nerica L44 can be one of the novel heat tolerant donor. So that let on this was because in the plant breeders also use that, it was recommended for the plant breeding, and this is the work of the plant breeders, and they have taken the Pusa Basmati 1121 is a well known basmati variety of India. So they wanted to, they have planned to introgress the heat tolerance trait of this variety NL44 into the background of the Pusa Basmati 1121, and you can see the how the in the normal conditions timely shown both the filled grain and unfilled grain. You can see the difference, there is no much difference, they both have the almost same number of the filled grain, number of the unfilled grain here. But under the heat stress late zone condition, this experiment was conducted by the plant breeders only. You can see the unfilled grain here in case of the NL44 is so small, while in the case of the this one, the filled number of filled grain you can compare, you can see the idea how much it's showing the sensitivity. So this line was introgressed by Pusa Basmati and NL44, and you can see we have, it is F80 stage, 20 cells are ready, and they are being QTLs are basically mapped for the reproductive history by Dr. Gopal Krishnan, head of the genetics. He is involved in this work as a collaborator. So this is the field work, which initially is work which we did in the field condition. Then again, because we had the facility for the exposure in the control environment, so after that we had taken another set of 240 genotypes, gone on the field conditions, and when the anthesis started, the booting stage, basically we transferred them, the individual genotype was selected, and at the booting stage they were transferred inside the greenhouses, and we focused on the primary tiller and the main tillers only. So we marked them to identify them, which are these tillers, to calculate the spikelet fertility. So the temperature inside the normal condition temperature was average, it was nearly 31, and the high temperature exposure inside these chambers, control chambers, was nearly 39. So this is the variability. This set again of the germplasm was obtained from the IARI. So you can see large variability was found. So we had the sufficient good time. So at one time the whole genotypes were not actually flowering. So we slowly when the flowering occurs, we shifted them inside the greenhouses. Then you can see the large how the variability occurs because of the high temperature effect. So these are the control one, and the purple one are the actually this orange color is the bars for the high temperature stress. What you can see here. So averagely what we found, the specular fertility and the heat stress averagely was 18%, while under the control is what 76% across the genotype. Now this is the data of the panicle granule. You can see the how the variation occurs because of the high temperature stress. And here you can see the average panicle yield in all the genotypes. It was 0.76 gram in the high temperature stress condition, while in the control it was 1.65. So it was nearly 50% reduction in the panicles yield itself, the weight of the panicle, because the grains are either they are not formed or they are not properly filled up. So this is again the percent reduction in the panicle yield. You can see large reduction is there. So most of large number of are having more than nearly 70% or more than 70% genotypes having more than 50% reduction in the panicle yield. Then this is the one of the physiological trait well known of the membrane stability. We are because with the work related to heat tolerance, we also analyze this one for the high temperature exposure. So we found largely the large variability was there, and this is the under the control conditions. It is having the you can see the nearly 65 or 70%, while in the heat stress condition it was less than 60%. Then the quadrant analysis was done for the specular fertility, and we found this is some of the genotypes which are having the green color. They were having the more than 70% fertility was there. Same way we also have the analysis was done for the panicle yield, and we found these genotypes which are maintaining the higher specular fertility. They also maintain the higher panicle yield. So these were they were. So from there we had taken the 6 of the contrasting type for the further physiological analysis and confirming them that they are having the really true tolerance for the heat tolerance. So we have taken some of the physiological traits like pollen viability and the oxidative stress level because of the accumulation of more H2O2, and these are the membrane stability in terms of thioreactive substances which indicate the level of the peroxidase of the lipid in the membrane. So you can see the interesting part is there this Nerical 44 is also maintaining high stability, and it is also comparable to the N22. Check. Same way you can look into the intolerant type, the pollen viability, how the sensitive type, the high temperature stress reduce the pollen viability. Also the level of oxidative stress was maintained because maybe due to the induction of the more intuitive system. So we had nearly the lowest level of the this H2O2 level in this Nerical 44. So these are just confirmation. Also we had gone for the thermal imaging to know the tissue temperature, and what we found that in the tolerant type the limb temperature was 3.5 degree lower than the ambient, and then the specular tissue temperature was also maintained to be lower side by 2.1 degree. On the other hand, we looked at the sensitive type, the temperature difference was depression was 2.4 and specular was 1.3. Then also the pollen germination, pollen viability is a separate trait. This is pollen germination stigma, the protein structure. You can see how the sensitive type, the number of pollen germinated is quite less compared to the tolerant type. So these are some of the physiological studies which we conducted to confirm that the line or genotype which were identified with the heat tolerance really represents the root of the tolerance. Then another study was that this was related to the story of the high temperature stress. Then we have, because the high CO2 is also one of the main, and it is known to be that CO2 can have the positive impact of C3 crop plants. So what we have done, we have used this methodology of the IARI. We have the set of the last same genotypes were there. So we screened them by using the this is called the low plant density. In the low plant density, basically, it was hypothesized that this low plant density may stimulate the response of the plant similar to high CO2 environment, just stimulation. And because both the treatment resemble the resource enrichment was there, that was the way basically. So what was done here, if you have the normal plant density where the between the rows the distance is 20 centimeter and the plant to plant is 15 centimeter, in the low plant density the between the rows was 40. So you can see the number of plants were nearly half of the optimum number of the plants. If the optimum we had 36, 37, here the number of plants were 15 to 16 per meter square area. So this way the between the plant to plant distance was also increased to 20. So that was the way how we this way was the pre-screening of the genotypes, that which one of them are heavy responsive. And this is the paper which was followed by Simono et al. In 2014, same methodology which was observed. So what you can see, some of the important traits like the SPAD chlorophyll values. Here you can see the LPD and low plant density genotype, they scored higher SPED, which indicating they are higher tissue nitrogen as well as chlorophyll content. So it can support them, then they can contribute to the better photosynthetic activity. Another was the flag leaf temperature. So in the plants which are grown the low plant density, they have scored lower flag leaf temperature or the leaf tissue temperature, and it is very well known that is related to the stomatal conductance and transpiration and also contribute to thermal avoidance. This was the some of the traits. Then if you look into the tiller numbers, when we have the resource enrichment availability, the treatment, so the tiller number is considered to be the main trait for this type of the study of the low plant density. So you can see how the number of tillers in the normal plant density and the LPD also, it shows the quite increase in the number. Same with the number of panicles were there. So we can say that the tillers are more under this conditions, and this is also the spread already I have shown to you. Then, sorry. So what we had done, we had calculated response index of them. Response index is based on the this is the formula given with the bill of metal for the stress tolerance. Same way we had taken the initially is for the individual genotype, and this was there. So what we observed, there was large genotype variability was there for the key CO2 responsive traits. And another interesting feature was that the elite and the high yielding cultivars like the Pusa Basmati 1121, which is very known high yielding variety, they responded better to this type of the treatment. So you can see this is the response index for all the genotype is scored, and some of the prominent variety like Pusa Rice Hybrid 10 and the Pusa 1121 Basmati, IR 64, also the N22 also have the in the middle size, but they scored better for this type of the treatment. And this is also the same way response index for the genotype for the number of tillers per panicles was calculated, and we found that the response for all the traits was found to be the same pattern. Then the this is the response index for the plant density for the grain yield, and also the same trend was observed. So after that we have calculated cumulative response index was calculated, and that was aligned with the grain yield under both NPD and the LPD. And the genotypes was categorized into three types: the low responsive type, the medium response type, and the high response type. So what we observed here, that some of the high yielding type, the PRS 10, also the Pusa 1121, they found to be representing the high responsive type to this treatment. And the NL44 and 22 type, which was having the high temperature stress tolerance, they also having the medium stress tolerance. So from here we have type. So because this was the method, it was the one of the pre-screening method. So we cannot exactly say that the response of the CO2 will be the same whatever it is coming. So from here we have selected the 23 contrasting type, which represented the low type, the medium type, the high type, and then they were validated. So from here just we concluded that the LPD can be potentially enhance the plant growth, that is the particularly the number of tillers can be more through the enhancing the photosynthetic efficiency or the resource availability. And it can be considered, this technique can be considered as a promising alternate option for the pre-screen response from the large set of the genotype. However, the confirming their response on the actual high CO2 element is also necessary for the selection of the candidate donors. So from here, as I mentioned to you, we had taken the 23 different, all representing all three types, we selected them, and they were grown under the actual CO2 element using the phase system, pre-enriched system was there. And the seedlings were transplanted and raised into 3 phase rings were there. The size of the each ring was 144 meter square. And the same genotypes were transplanted, which were taken in the open field condition in the open field as well as the low plant density. So the normal control was the normal plant density, and the LPD and the phase were the two treatments were there, and we wanted to compare. But the response, what was there in the earlier explained, the low plant density and the phase, how much they differ, whether they are similar or they are varying here. So here, what we observed here, the photosynthetic activity, because when we talk of the CO2 response, always the photosynthetic rate is the most responsive trait here. So what you can see that in all the 23 genotype, basically the rate of photosynthesis increased. But some of it varies among them, but compared to the control, even in the LPD conditions also, low plant density also rate of photosynthesis was higher. So it was, and then we calculated the photosynthetic water use efficiency as a ratio of your stomatal conductance with the photosynthetic rate versus the stomatal conductance, and that was found to be the photosynthetic water use efficiency. So it has also found that this photosynthetic water efficiency also improved because of the higher photosynthetic rate in most of the genotypes, but some of them they have the reduction depending on their response. So this was the photosynthetic response, but when we compared the agronomic traits, This is the percentage increase over the control in the low density type and also the phase system. What you can see in the number of tillers, in the low plant density there was sort of nearly this average of all of them 57% was there, but in the phase system only 44 system when the actual CO2 environment was there. Same with the panicle number, we observe the same trend. The panicle weight and the total biomass and the gain yield, you can see gain yield showing in the LPD it is showing 41% increase, but in actual CO2 environment it is only 17%. Same way with the panicle weight 38% here and the 25% was there in this one. So what we concluded from here that all 23 responded to high CO2 in the phase system, but the magnitude was lower than the low density type. So the main inference here is that for optimum response of the high CO2 if you have to take, we have to look into the other factors like the light condition and the nutrients are also important. So if these factors are also at par with the low plant, then you can have the CO2 real response is there. So after that one, this is again the cumulative response was there, so we can find the large variability among the some of the genotype that responded. These are all 23. So this was they showed the variability under the low density response, this one. And then we also comparative analysis was done for under the low plant density phase, we found the good correlation among them. So this was said that the rise section, so this is a significant correlation was found between the cumulative response index and the rise section under both the conditions. So we have that was the conclusion that really it can be the until unless we confirm the CO2 response. So from here we can identify these genotypes can be further validated and they can be used also for the plant breeding program along the heat tolerance. That was the main conclusion. From here, lastly we have taken the two contrasting genotypes from our study in the earlier one, and then they have grown the open top chambers, open side, because here the temperature conditions in the phase system it is difficult to create. So in the open top chambers we can create the high temperature as well high CO2. So the two of them, Nerica L44 which was there identified the donor in the first slide, and also the BB1121 were taken and they were raised in the open top chamber. CO2 exposure was started from the panicle initial stage and continued to the physiological maturity, and but the high temperature and combination of the high CO2 temperature were given the heading to maturity, because as I mentioned the stage of the high temperature sensitivity is always the reproductive stage. So we started from the heading onwards till the maturity is there and other traits were analyzed. So what you can see, then when we saw the rate of photosynthesis here in this Nerica L44, the CO2 response is quite positive for the photosynthesis, and when the temperature was there it is showing the as such the complete when CO2 and temperature was there it was reduced because of the high temperature, and when the only high temperature was there the rate was further low. It shows that despite of the tolerance to high temperature the photosynthetic activity is affected, but if you compare the Pusa Basmati variety sensitivity type, you see the reductions were large compared to them. So it shows that photosynthesis system is really very sensitive to high temperature stress, and that the sensitivity can also be related with this photosynthesis rate. Again the stomatal conductance, the response was quite differ. Very interestingly, under the high temperature the conductance is reduced because stomatal regulation is very sensitive to the CO2 level, but in this case under high temperature conditions we in NL44 we found the higher one, the conductance was induced, and that can contribute. Basically this is related to the cooling capacity of tissues. So this can be one of the trait which is contributing for the heat tolerance of this genotype. But if you compare the another one sensitive type, you can say here that the response was quite low. So we can say that one of the reason why the Pusa Basmati is showing the sensitivity is the region that this stomatal conductance is basically not induced under the high temperature stress conditions, so that it can raise the tissue temperature that can be affected the photosynthetic activity. Another interesting part is the metabolic of the carbon metabolism. So we have measured these are the data in the basically flag leaf during the photosynthesis. So we have taken the sucrose synthase. Sucrose synthase basically reflects the availability of the partitioning of the sugars from the photosynthesis to the growing sinks here, and then the ADPG pyrophosphorylase and the starch synthase, they are the very important enzymes involved in the starch biosynthesis. ADPG pyrophosphorylase basically it is involved with the biosynthesis of the substrate for the starch synthesis, there is ADPG glucose, and starch synthase is also very important enzyme for the starch synthesis. So we have seen here So what you can see that once we have the sucrose synthesis was measured in both the genotypes, and the CO2, the response was almost same because of the CO2. Then again, when the only CO2, when the CO2 plus temperature was given and only temperature was given, this variety did not so much change in the sucrose synthase. So it shows that this variety may be basically here it can be supporting the availability of the carbohydrate for the growing seeds or the seed development with the for the starch synthesis. But in case of this one 1121, the partitioning of the sugars towards the by having the lower activity, we can say that this is affecting the availability of the sugars for the grain development. Another way, so this is again you can see, but the response of the sucrose synthesis enzymes ADPG pyrophosphorylase as well as sucrose synthase in these both the varieties, here you can say there was no effect in the CO2 as well as this one. So it shows that this process is very sensitive for the grain yield, the rice, and here this was not affected, and it is showing that really it is tolerant for the high heat stress for this enzyme activity, which is also very sensitive. But if you compare the CO2 response for this one in this variety, it is among both the for both the enzymes, this PUSA 1121 is supporting the activity is very high because of CO2. So CO2 response is highest, but under the high temperature conditions, you can say it is also reduced to nearly half of the CO2 response, and also same way here. And in this variety compared to the NL44, this CO2 when it was combined temperature CO2, it was not able to basically negate the beneficial effect, was not able to combat completely. So it shows that here very clearly you can see this can be one of the reason how the grain yield can be affected, because it's contribute to the grain filling stage a lot. So then you can see here how the shoot biomass was there, because of it can affect the grain yield and the grain weight can be there. So seed set, because of the availability, seed set or the fertility is the same thing. So the fertilization might not occur, so it may be less fertility can be there, less seed set be there. So compared to the tolerant type NL44, this one it is showing the very high sensitivity to this stage. It shows that this really the main reason could be the availability of the carbohydrate at the time the seed development stage. That can be the main reason for the sensitivity here contributing. Then lastly, you can see the same way we found the effect of all the stress treatment, the CO2 as well temperature here in this one compared to the NL44. So very clearly it shows that what could be the reason for the sensitivity of PUSA basmati 1121 for the temperature stress, and CO2 was not able to mitigate the high temperature stress in this variety. So that could be one of the reason, while here in case of NL44 there was not much effect because of this one. And the grain yield you can see it is also reduced nearly to the half. So with that one we could conclude that the high CO2 can enhance the photosynthetic carbon assimilation and increase the temperature beyond the 20 degree and can negate the positive response of the CO2. Large genetic variability exists in the rice accessions for high temperature stress tolerance and also the CO2 response. So developing high temperature tolerant varieties can be the best approach for the climate resilience in the future program. And if we pyramid the traits of the high CO2 and temperature together, that can be really the one of the best approach for developing the climate resilient rice genotypes. So this is my summary of this presentation, and for this one I would like to just give a thank to my postdocs and the SRF who contributed for this one. And before ending, I would like to show you just a glimpse of this facility, which is one of the new state-of-art facility has been developed with the grant got from the Indian Council of Agriculture Research. Phenomic facility is there. Professor Deek visited, and all students have also worked, both SNA and all. And this is the system you can see. We have the RFID tagged, chip tagged ports are there. They are moving in the conveyor belts, and then these are the imaging platforms. So right now there are the 8 chambers are there for the imaging, but 6 of them are working perfectly at this time also. So overall summary of this facility is that we have the 4 high-tech chambers are there, where we can have the control conditions for the temperature, light, the RH and the CO2. Then the moving conveyor belt is there with the platform of 1200 RFID tag brands carriers, and they also have the 5 stations for the automatic imposing soil moisture stress, measuring the evapotranspiration and the water use efficiency. And these are the list of the 6 sensors which are used, and we are using for the analysis of the data, the Amnitech system is being used. So with that, I thank you all for your good patience, and now I am ready to give you any queries if you have this one.
Thank you, Madan.
Yeah, I hope I covered
some questions for Madan. He rushed through a lot of data, but I am sure some of you. First Shen, then Neil.
Okay,
And
I'm Shane Chen from the UWA Institute of Agriculture, working on heat tolerance in Kelowna. Thank you very much, Professor Singh, for your wonderful talk. And Regine, at first you analyzed combined high CO2 and temperature response in 71 rice lines, and using the field-based open-top chambers. So I'm particularly curious about these open-top chambers. I wonder how much higher of CO2 and temperature you can achieve, because you mentioned, for example, 35 degree, if it can be tolerant, you can take as heat tolerant.
Yeah.
So can you achieve 35 degree? And in this case, what's the temperature beside the facility?
outside the facility.
Yeah,
Yeah.
outside the facility. And also, how long can it last in the field condition? That means how many hours you can retain this temperature? Can you please
Yeah, yeah.
explain more?
Basically, two types of chambers were used. First, I will talk to you about the temperature which is used for the increase the high temperature exposure for the rice in the field conditions. Those chambers were basically closed from the top, and when the temperature outside was nearly 32 or 30 degrees Celsius, by trapping the poly cover chambers inside, we put our data loggers and also monitored the humidity, and that inside the temperature was 36, and because of the warming, you can see the greenhouse effect of the poly chambers inside, and they are closed from the top, so you can have 5 to 7 degree temperature very easily. And if it is going to be higher, sometimes when the rays are very high, when sunshine is higher, even it can be more than that. So we have to put the pores inside the poly chambers, and we have to release some air so that the temperature should not go beyond 38 degree. Otherwise, it will be lethal to the plant growth itself. Second, open top chambers, which I have shown the end one, they were the open top, means they are having the circulation of the air continuously from down to the up, continuously air is circulating. Only the air from the lower side, for when the air is entering, we are having the basically the CO2 plus air is mixed together, and that is blown inside the chambers. So when the air is circulating continuously, at the time we are thinking that only the CO2 is changing, other conditions are not changing. For putting the CO2 plus high temperature, we have to use our blowers separately, because under the open system condition, it is really not very easy to create the high temperature. So we have to use our thermal heaters are there, and then also need to be regulated. Then only you can have the both CO2 plus temperature. So both are differently used.
So the continuous flow.
Yeah.
So maintaining that, otherwise.
So in the field rice system, they are fixed only at the time of the flowering, not continuously. But the open top chambers are fixed structures inside. They are fixed there. We can grow different types of crops.
Neil and then Nanti.
Very nice seminar, by the way. But whenever people work on heat tolerance, the one thing that worries me is whether you have also maintained the water status of the plant. And I presume your rice is irrigated, because rice
Completely,
is irrigated.
yes, yes, yeah.
But that still doesn't mean that they're not subject to some water deficit during the...
Not at all,
when
yeah.
Well, have you checked that? Because where stomatal conductance is lower, and that's an indicator to me that you have a drought
Yeah.
stress coming in there. That's why I asked
Very
the question.
important, yeah, very important observation you have made. It is really very interesting. Even we were also surprised. First one, whatever pots we have grown for the rice, they are flooded water basically. Every day the water
That
is...
could be a problem.
Yeah. So when these chambers were there inside the grow chambers basically, but the water was, scarcity was not there. Only the humidity was changing, so it can be because of the humidity conditions are changing inside. And for rice, the humidity is very high. So in the grow chambers, the temperature as well as humidity was also higher. So that high humidity can be affecting, could be the one of the region that can be having the effect on stomatal opening. Then can be.
The temperature per se may have some effect on the stomata, overriding the water. Because the tissue is getting really, I mean, this is what happens in Nile Valley, Nile Delta, when you have plenty of water, but the heat stress 45 degrees centigrade can
Yeah, absolutely. That's why when it's flooded, it can also be a problem.
Anyway, one thing, good question. Now, Nanti,
thank you for the presentation,
Merci.
great presentation.
Yeah.
I'm not a gene jockey like you or some other people in the front row. I'm a down-to-earth soil scientist.
Good.
So my question will be very general. Now, Professor Sidek just published a paper. The best approach to sequester CO2 is to grow more biomass.
Yeah.
And that eventually leads to more soil carbon, what we call recarbonizing soil.
Yeah.
That's what my area.
All right.
And so my question to you is, I understand when the CO2 concentration goes up, invariably you produce more biomass.
Yes, yeah.
So therefore, by producing more biomass, you are sequestering more carbon.
Yeah.
So is this positive feedback eventually will help to reduce the CO2 in the atmosphere? That's my question.
Yeah, that can be really, because the forest is must have read the literature. The forests are being used in the different places to sequester the CO2. Same is because they are the perennials. Perennials are the best source to sequester CO2 that we use. The plants are annual, at least you can have not continuously not be there, can be there. So they are basically that is the only way.
And there is a lot of data, for example, if you take the Indian data or Chinese data. So India produced about 382 million tons of grain. Once upon a time it was only 50 million tons of grain. Of course, used more fertilizer, etc. There is a lot more CO2 is captured. The argument is that a lot of that carbon either temporarily stored in the soil, especially under the roots. The same with the China. If you take a harvest index of 40% and with a yield of 6 ton rice and so on. So there's quite a lot going on, but the problem is how long we can store, etc. But I got a question. I did not fully understand, again, excellent talk, as usual. I did not understand the low density situation. So the low density, basically you're saying that you have not applied any CO2.
No.
But the low density, the problem for me is that the single plants got more space,
Yeah.
they will till more, and they have got more nutrients available. I still don't understand how you relate that with CO2 fertilization effect. I didn't
was the simulative effect, not the CO2.
Yeah, but I still don't understand what it means. I don't know.
The people.
I'm slow learner. Yeah? So tell us what is that? I know you read that. Explain that.
What Simonov group has basically hypothesized, they said the way when we are giving the CO2, it is responding because the more photosynthetic activity is there, more carbon assimilation is there.
Sure.
So same way, when only CO2 is not there, but the other nutrition factors, light conditions are giving more to the way the CO2 is responding to the plant growth and biomass, and the same proportionally here, if the CO2 is not there, but more light, nutrients and water is available, so it can have the same type for the pre-screening. In fact, not like the CO2. Yeah. So enrichment, like the enrichment of CO2, here the enrichment of the other nutrient factors is there.
Okay. But my problem is that you looking at an individual plant,
Yeah.
and let's say only 4 plants here, and you got individual plants.
Same
very
way,
good yield,
yeah,
which is
yeah.
the more yield, you get a total yield, let's say it's 10 grams. And I can pack more plants per, an individual plant will be less, but I can get 20 grams in that. So that density factor, I need to look at that. I haven't looked at that literature, but I don't want to have an argument, but I'm saying I don't understand that. Sorry, at the moment. Yeah?
Yeah, that's I agree with that.
Now,
Yeah.
You first.
Hi, Doug Hall. I'm an agricultural policy person, but I'm a plant biochemist by training. What I'm interested in is the policy implications for your work and also the reception that it's received, because my experience here is that when this current state government developed climate policy, its presentation about agriculture was negative, 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 no positive aspect at all. When I challenged them on the obvious benefits of high CO2 to C3 plants in particular, you treat it like a heretic and basically dismissed. I mean, to me, what you're doing is engineering the genes that existed in geological plants when there was much higher temperatures and much higher CO2. And clearly plants did survive well in those conditions. So I'm just interested in how your work is received in policy areas in India and
No, this is not. We are not involved on this.
Yeah, I think the answer is that the government of India has spent a huge amount of money. The facilities, the phase facilities, the phenomic facility, all were created, the current Director General of ICAR, a great man, to study the interaction between CO2, temperature and so on. So India is leading that. The second thing is that India in the G20 and COP meetings, etc., has been criticized about not taking enough action for reducing the greenhouse gas emission. Of course, we can argue per capita emission is very low in India, but the number of people, 1.48 billion people, the same with the China. Whereas our Australia per capita emission is very high, so the total emission is not big. So the India government is taking this very seriously and getting data generated within India rather than depending upon European data or North American data, number one. The same here in Australia. Louis Barton and others from this university done some wonderful work and showed that the emission of greenhouse gas from our dryland cropping system is substantially lower than the European data. And our greenhouse gas commission agency changed, because in Europe, you got the summer rainfall coming, huge amount of activities going on, huge amount of CO2 and everything coming. So the government of India is trying this kind of work to show that, not necessarily this
But
particular.
irrespective of what happens to global temperature
Yeah.
in the future,
Yeah.
the work you're doing, and you gave some good examples in Africa, is going to extend the agronomic territory that species can be grown in, and that's got to have great developmental benefits.
Absolutely, yeah, true. You, go ahead.
Thanks for the presentation. I just got one question. When you try to do the screening genotype of the rice under both heat stress and also high CO2, did you consider or have you checked the stomata characteristics? Because it's, in my understanding, like either the stomata development or its characteristics will influence the water evaporation and also control the leaf temperature. Maybe contribute to that.
You mean stomatal structures?
Yes.
Yeah, like density.
No,
Yeah,
that
They did not. They did not.
not.
They
It is
looked at the
yeah.
conductance and.
Because we have the large number of genotypes are there, and we non-invasive
Maybe a
phenotype.
PhD student.
Yeah,
can
that can be
that
there.
work, and-
Yeah, not
But
focused.
I have one more question is about, it's lost now. Talk about the temperature. Look, this basmati rice is adapted to low temperature, because if the temperature is up,
Yeah.
the basmati aromatic
Really.
flavor is gone. So I'm not surprised the decrease in all activities you mentioned with high temperature. Whereas your NL line is coming from Africa where the temperature is high. So I'm not surprised, that's good work. But when you put that line into the basmati, there would be a drag and you are going to lose the
Quality.
aromatic
Yeah, quality.
quality. So I'm sure in breeding anything can be done, that's what Wallace says. But that's my worry, because this is coming from the New Africa rice, which is really high temperature tolerant, so as we can take rice from Kerala or somewhere and put it in. But the basmati, as soon as put it in, then you get the drag. Also, you take basmati rice from those belt to another area, then it won't show the flavor.
Why?
It is to do with the temperature. They are really sensitive to temperature and flowering and growing season, etc.
But these are
This is why Punjab province in certain parts in Pakistan, that's a basmati growing belt. Delhi is fortunately a good.
In
basmati
Punjab
belt.
also, many times now the farmers are complaining in many seasons. When the temperature high coinciding with the flowering stage, it is affecting the quality as well as quantity both.
really.
Basmati. This is the main.
Yeah, my feeling is
Empty
that
fields are there actually sometimes. Yeah.
my feeling is that it may be even pre-flowering stage.
Yeah.
Wallace?
Yeah. Maybe that is the.
So that raises one more question about quality in grains that are harvested under elevated CO2.
Yeah.
Have you measured the components and nutritional value of the grains under elevated CO2, and do you see the reduction in protein? That
Yeah.
is
My,
obvious?
my colleagues have done that work in wheat as well. They have worked for the CO2 also particularly, and the not only the reduction in the protein level, but also some of the nutrients were also losing. Important nutrients were also lower. Yeah, they found the declining, and the regions mostly they claim to be the dilution effect because of the more carbon assimilation, and so it was recommended by some of the scientists and to maintain the same pace of the gain yield and the quality, we should have the recommend the application of the additional nitrogen for the protein quality.
I have a good breeding method for that.
Okay.
But I think
That could
we had a review that was published on that on wheat.
Yeah.
Again, the temperature is going to down. CO2 by temperature effect, yield will go up, but the quality will go down. And the same with the pulses, it's a big issue. But as Madan said, then we may need to have more, particularly nitrogen.
Yeah.
Then what will happen to the water?
Yeah.
Because in our case, temperature by water stress by CO2 interaction.
Yeah.
One of my PhD student, Eduardo,
Right.
does that in wheat, and he did some wonderful. It's quite complex, and as you get it, there are 3 interactions, and the temperature goes up, yield and everything goes down, despite you have the CO2 double. Any other questions? Yes, Nanti.
Just following on Wallace's question, one of the issue with rice is zinc nutrition.
Yeah.
I think that's becoming a very critical issue. So most of the Indian soils are zinc deficient.
Now the lot of fertigation work is also going on for the zinc.
Biofortification.
Yeah.
So
Yeah.
that's what my question, you are correct.
Yeah.
So I'm just wondering, by increasing the yield, whether it is dilution effect or some other effect, do you think the nutrition quality, especially in relation to zinc, will have some impact?
About the effect on the high yielding variety, I don't have much idea, but our scientists have been really planning to develop the biofortified varieties for additional zinc amount with the high yielding. But whether earlier one, high yielding one were having the low zinc, that information I really not having the idea. But now they are fortifying, we are developing the varieties with the more zinc content. That is work is going on. Even for vitamin A also. Same way like the other people done, the vitamin A work is also being done.
Golden rice.
Yeah, golden eyes. Yeah.
But our review on wheat clearly shows that zinc and magnesium, these are all going to be affected by if you do status quo.
Yeah.
All right.
Thank
you very
Okay,
much,
yeah,
Madan.
right,
Just before I closing the seminar, this is already recorded in full or will be recorded. The video will be uploaded to UWA Institute of Agriculture YouTube channels this afternoon. Then we have some upcoming events. The QR code is there. The first one is on 28th September, Professor Paul Kenyon from Massey University, New Zealand, for our 2023 Alan Xavier Memorial Lecture. And Paul will be delivering his topic on improving per ewe performance. This is on sheep, the driver of profitability in New Zealand flock, because Alan Xavier lecture is must be on animals. So he's coming. Unfortunately, I'll be somewhere, but others will look after that day. And next month, I think that's on the QR code. Next month, on 6th October, we have a very exciting event, our 2023 UWA Ridgefield Open Day. The Open Day will feature various demonstration and showcasing some of the best practice farming system. So we'll have buses going from Crawley. I think 3 buses have been booked. And I was told that Vice Chancellor is coming. Some Senate members, Deputy Vice Chancellor and others are coming. So it will be a good day. Your lunch will be provided. You just come, make sure that you are registered, and the bus will drive and then take you, and then you will be brought back to the Crawley campus. Particularly students, if you want to know more about, the details are there on the QR code. Finally, probably will be our last for this year, hosting the 2023 Hector and Andrew Stewart Memorial Lecture, which you all know that it's a very popular lecture. And I'm pleased to announce that we just got the information. Adjunct Professor John Dixon, you all know John Dixon. John Dixon has worked in the international agriculture research on sustainable intensification. He worked in CIMAT and CGIAR centers and so on. He's an adjunct professor at the University of Queensland, and he's agreed to come, and we will have more details, exactly the lecture and details. So he's doing well. So thank you once again for joining us, and good afternoon. If anyone wants to meet Professor Madan Pal, just contact Cora. He's still around until even next week, but I'll be away. But so you have an office and you can meet Madan. Thank you very much again.